So Jen, can you go to the society page? Cool. I'm sure a lot of you are familiar and have joined one of our talks before. We are a nonprofit society, a community of scholars or ex-scholars who are very excited about science and technology and are hoping to learn more about, the most cutting edge research, to meet new people and to share our exciting ideas with each other and even share our ideas outside of academia. So our society has different branches of talks and activities. The core, which is the one you're attending today, is focused on academia. So every week we invite speakers to introduce their research, and we're not restricted to one or two different fields. It's, we're open to anything. And the whole idea is for people to learn about things outside of their immediate research project. We also have non-academic talks, so people who used to be PhD, like myself, but who decided to find a career outside of research, and we're coming back and sharing with people our experiences, what are the insights, including, if you're keen to translate your research. We had a talk on tech transfer by Hyungwoo, as you can see on the poster. We also have special sessions like skill set sessions that are pragmatic and helps you, with skills like figure making, for example, the one that Jen hosted last year. We also have special issue talks where we invite very prominent figures in different areas. For example, Professor Li Tong, who is a very famous scientist in the US. And we also had comedian Huang Xi give a talk about his experience from academia to comedy. So this is basically an overview of our activities. And again, welcome you to this session. And please, if you haven't followed us, follow us and stay tuned for the next couple of events. And now I will hand over to Hakan to introduce our speaker today.
Thank you, Bihe, and thank you, Jen, for giving me the opportunity to host today's speaker. So hi, everyone, and good afternoon from Germany. So I'm Hakan Ceylan. I'm currently a senior scientist at Max Planck Institute for Intelligent Systems. So we are based in Stuttgart, Germany. So my research focuses on developing robots at the size scale of a single cell, which is around 20 micron. So our goal is to use these tiny robots to leverage minimally invasive interventions and targeted therapies. So before introducing the speaker, a very quick announcement I would like to make about my changing situation. So I will be moving to Mayo Clinic next year to start my independent group there. So it will be a multidisciplinary translational research group. So, and there will be multiple openings. So if you check my research from our institute webpage, and then if interested, just drop me an email, and then you'll see what we can do. So, yeah, keeping this part short, I would like to introduce today's speaker, Dr. Kojin Chen. He's an assistant professor at McGill University. So there he's affiliated with a biomedical engineering department and Rosalind and Morris Goodman Cancer Institute. So Dr. Chen received his bachelor degree in chemistry and PhD degree in material science. So before joining McGill, he was a postdoctoral researcher at UCLA. So Dr. Chen's research is super exciting. It focuses on engineering novel biomaterials and devices for drug delivery in the context of precision medicine. So he's an expert in this field with over 60 research papers and applied patents. So as such, one of his patents has already been licensed and has been accepted into FDA's emerging technology program. So yeah, let's see what Dr. Chen has to say more about more on the recent developments from his labs. So Dr. Chen, so stage is yours.
Okay, thank you very much, Hakan, for the nice introduction, and congratulations with your coming independent research program.
Thank you.
So can I share my screen right now? I think you have to stop sharing first. All right. Can everyone see my slides? Okay, great. So again, thank you for the nice introduction and for the invitation. So good morning, everyone from Montreal. So it is my great pleasure to be here and give a talk about my research. And today I would like to share with you some of sorry. So my slides is still okay, right? Okay.
Yes, we see.
So today I would like to share with you some of my research projects, which mainly focus on nanoformulations and microdevices for drug delivery. So obviously, the first question we want to ask is why we need drug delivery system, right? So brief answer is we all know that drug are being used to improve our health and extend our lives, and we know as well that most drugs, even those discovered use the most advanced technology, they have an unacceptable side effect due to their unwanted interactions with with healthy tissues. And clinicians are doing their best to direct the medications to the disease sites, but oftentimes it's very challenging and they fail to do so. So the lack of this precision limits our ability to design optimal medications for many diseases. So the big question here is a bioengineering. So how we can use engineering approach to deliver drugs to the sites of interest and to achieve this precision medicine. So like a Uber driver, right? So hopefully we have advanced tools like smartphone or GPS to drive our passengers to their destinations and to achieve this precision medicine. So drug delivery is a very active field, and advances in biomaterials design as well as micro technology offer the solutions to achieve this precise drug delivery. So these two types of drug delivery systems that are totally in different scales. For example, the nano formulations that are in the nanoscale and the micro devices that obviously they're in the macro scale. So till now, bioengineers are still optimizing these two systems to achieve this big goal, the precision medicine. And here I would like to share with you how I explore and engineer these two systems to deliver drugs for improved disease treatments. And I will start with nanoformulations to deliver drugs, including small drug molecules, nucleic acids, and genome editors. And I will then introduce a micro device-based drug delivery system, especially for cancer immunotherapy. So let's begin the first part of my presentation, nanoformulations for drug delivery. So drug nanocarriers actually have been investigated for many decades, and they have many advantages, of course. So for example, by encapsulating the drug inside the nanoparticles, so we can improve the stability and the solubility of many hydrophobic drugs. And we can also improve, and by using the nanoparticles, we can prevent the drug from degradation. And nanoparticle can also, depending on the design, so nanoparticle can offer controlled drug release and provide specific cell tissue targeting capabilities by functionalizing the surface of nanoparticle with certain targeting ligands. And also we can incorporate multiple type of drug payloads in one nanoparticle system to achieve multifunctionality. So because of these advantages, so drug-loaded nanoparticles have been used to treat many diseases such as cancer, infections, and also they have been investigated for regenerative medicine. So despite a lot of active research in this area, so the clinical outcome of nanomedicine, the drug nanocarriers, actually remain a bit disappointing. So particularly in the field of cancer treatments. So although numerous efforts have been devoted in this area, so there are only a few approved nanomedicine for treating cancer. So of course, there are many reasons. From the clinical and the research results, so we found that one of the major issues associated with conventional drug nanocarriers is their poor in vivo stability. As you can see here, that the many conventional drug carriers are formed by self-assembly of multiple polymers and drugs through relatively weak physical interactions, for example, the hydrophobic interactions and the hydrogen bonding. So these interactions are generally weak compared to the covalent bonds, right? So once these nanoparticles are injected into our body, so in the bloodstream or in the extracellular spaces, there are many factors that can disrupt these nanocarriers and cause the premature release of the drug before they reach to the target sites. And for example, here is the failure of this nanomedicine called NT911, which is self-assembled nano drugs. In the failure of this drug in the clinical trials is because that the 50% of the drug released from the nanoparticles in the bloodstream within 1 hour. And another reports also indicate that 80% of this self-assembled nanosystem dissociate quickly in the bloodstream. And there are certainly many other reports indicating this instability issue associated with the conventional nanocarriers. And while the purpose of using or designing the drug nanocarriers is to deliver drug to the site of it, to the target site. So if they dissociate before reaching to the target site, so these carriers basically lose their function. So my research aimed to address this instability issue. So I focus on designing stable nanosystem for drug delivery. So here I would like to introduce two stable nanosystems that I have been studied and worked on. So one is called Unimolecular Nanoparticles, and the other called Nano Capsules. So although they look quite different in this schematic picture, so the common thing they have is that there are only covalent chemical bonds within these two systems. So as I mentioned, so the covalent chemical bonds are more stable and more robust compared to the covalent physical interactions. So by using which means that by using this design, so the nanosystem are have a superior in vivo stability. And by using the right chemistry and polymer design or nanoparticle design, these two systems can be used to deliver various type of drug payloads, and for example, small drugs to the big biomicromolecules. And I will elaborate them separately with the unimolecular nanoparticle system first. So unimolecular nanoparticles, or UNP, so literally means that one molecule forms one particle. So the molecule is often called multi-arm star block copolymer. So you can see that. So here is the schematic picture. So you can see that you can imagine the structure is like the puffer ball, the toy that having many arms on the surface. So no matter how we play with it or squeeze it, so so they remain their original ball structure. So compared to the conventional nanoparticles, so I will introduce the specific UMP system in the next slides. So compared to the conventional nanoparticles that are formed by multiple molecules through physical interactions, as I just mentioned, so there are many factors in the bloodstream that can dissociate this conventional nanoparticles, like dilution in the bloodstream, the flow strength, the interaction with serum proteins, as well as the changes in pH and ionic strength. However, unimolecular nanoparticles, because of the covalent nature, so they can remain intact in the bloodstream, so enabling its superior drug delivery efficiency. So this is the basic structure of the UMP. So basically, it has this green central core to link these arms covalently together. So the core can be hyperbranch polymers, dendrimers, or even some small inorganic nanoparticles. And this red part is the functional inner block, so which is the key region for drug encapsulation. So it can be either hydrophobic or hydrophilic polymers, depending on the nature of the payloads we want to deliver. And I will elaborate this in the next slides. So the blue part is a hydrophilic shell. So typically it is polyethylene glycol or PEG, which is often used in the field of drug delivery to make the nanoparticle water soluble, and also to reduce the nanoparticles interaction with the serum proteins. And this PEG polymer can also allow us to conjugate functional molecules like target ligands or the functional moieties like imaging probes to achieve the multifunctionality of this UMP system. So as I mentioned, so this red region is very important for drug loading. So it can be either hydrophobic or hydrophilic. So for the hydrophobic drugs, like many chemo drugs are hydrophobic, so we can select hydrophobic polymer as this inner block to encapsulate those drugs. And the loading mechanism is mainly through the hydrophobic interactions and hydrogen bonding. So for the hydrophilic drugs, oftentimes they process like the nucleic acids or peptides. So oftentimes they process certain charges on their surface, so either negative or positive charges, right? So then we can design the polyelectrolytes to load these payloads through the electrostatic interactions. So I will give one example for each type of inner block, and the first example is to use the UMP to deliver hydrophobic anti-cancer drug to treat a cancer called triple negative breast cancer as an example. So very brief introduction of triple negative breast cancer, which is a more aggressive type of breast cancer. It's a subtype of breast cancer, but it's more aggressive and cause many breast cancer related deaths. And clinically, there's no targeted therapy for triple negative breast cancer. So although there are several drug candidates, for example, the Immunotherapy, which is a hydrophobic anti-cancer drug, and it also shows pretty good growth inhibitory effect towards the triple negative breast cancer cell lines. However, it has severe systemic toxicity to the healthy tissues, mostly to the lung tissue, which causes the termination of their clinical trials. So obviously, to rescue this drug for triple negative breast cancer treatments, so we need to find a targeted delivery approach to avoid or to minimize this toxicity, and also to potentially further enhance its therapeutic efficacy. So the UMP system is perfect for this application because, well, it's a stable non-platform, right? And it can also deliver hydrophobic drug. So the last missing piece is the capability to target triple negative breast cancer. So I found that the triple negative breast cancer often express a very high level of EGFR, which is an epidermal growth factor receptor on their surface, and a short peptide called G11 peptide can bind to EGFR receptor effectively. So that's exactly what we need. So here comes my design to deliver this immunoflavin drug for triple negative breast cancer. So this is the basic UMP structure, and the hydrophobic region here is made of polylactide or PLA, which is a very compatible and biodegradable polymer, and also is being FDA approved for many biomedical applications. And it formed this hydrophobic region to load this drug immunoflavin. And we also have G11 peptides on the surface to achieve active targeting capabilities. And here is the TM image of the UMP. So you can see that it's spherical and very uniform. And this is the enlarged picture. So it's showing the nice quadrilateral structure of the UMP. And while the most important thing we care about is the target delivery efficiency, right? So we test the target drug delivery efficiency of this platform in the mouse that have triple negative breast cancer. And 4 hours after systemic injection of the nanoparticles, so we measure the drug concentration in the tumor. And we are quite excited to find out that this platform, so without the target ligands, actually this non-targeted version can deliver the drug 7 times higher than compared to the drug that without any carriers. And more importantly, with this G11 peptide, this targeted version can deliver 72 fold more drug to the tumor sites compared to the free drug. So, which indicating the superior drug delivery efficiency of using this targeted version. And more encouragingly, so we found that this AF-loaded targeted version can actually lead to a complete tumor regression in this mouse model. And the dose I want to point out is that the dose we used here is only 7 milligram per kilogram, which is only like around 10% of its therapeutically effective dose that were tested in the clinical trials. And by using this very low dose, we can achieve this very nice tumor regression in the preclinical mouse model. And moreover, we did not identify any toxicity sign during the treatment or after the treatment, as measured by the body weights, as well as the histological analysis on the major organs like the lungs and liver and kidney. So overall, in this project, I use this UMP system to deliver hydrophobic drugs to treat triple negative breast cancer, and in a targeted manner. And of course, this platform can be also used to deliver other hydrophobic chemo drugs to treat other type of cancer. And however, while one thing to note is that cancer are quite different in nature, as you know, and we still need to do some research to understand the background and characteristic of each cancer and the payloads before we can come up with a appropriate delivery system, like the choice of target ligands, the polymers, as well as the dose of the drug, etc. So I just show you the delivery of this hydrophobic drug using this platform. And now I will give a quick example of using a hydrophilic polymer as this inner block to deliver a hydrophilic payload. And I will use siRNA as an example. Oops, okay. So siRNA, a brief background is it is known as a silencing RNA. So it's a short nucleic acid, like 20 to 27 base pair in length. So it can temporarily silence the expression of certain proteins by degrading the specific mRNA in the cells. So it has many therapeutic applications for disease treatments like cancer, neuron-related disease, or liver-related disease. And it while the the question or the limitation of this siRNA therapeutics is that this molecule is a very negative charge molecule. So the cell membrane is also negatively charged. So siRNA cannot effectively get into the cells by itself. So that's why it needs nano delivery system to deliver the siRNA into the cells to silence or to degrade the mRNA. And also the biomicromolecules, many biomicromolecules are labile or unstable in vivo because of the presence of many enzymes or nuclease, etc. So the siRNA and other biomicromolecules, the delivery vehicle is very important for these payloads. So here, because of this negative charge nature, so I designed a UMP system with this positively charged polymer as this inner block. So here is the chemical structure of this positively charged block. So it can complex with the siRNA through the electrostatic interactions and form this stable siRNA loaded UMP UMP system. And the UMP can get into the cell through a process called endocytosis, while the cell membrane engulf the nanoparticles and form this endosome inside the cells. And the endosomes were further fused with the lysosomes. So these compartments are acidic, and also it has many. So these compartments have many enzymes in these compartments. So so the siRNA, if it stay in this compartment, siRNA can be quickly degraded. So we need to help the nanoparticle escape from the endosomes or lysosomes. So here we integrate two moieties in the nanoparticle design, two pH responsive moieties in this nanoparticles. So the idea is like these two molecules can induce the damage of endosome membranes through a proton sponge effect, where the protonation of these two groups can induce a osmotic pressure inside this compartment and lead to the membrane disruption. And this facilitates the endosome escape of siRNA complex. And once in the cytosol, so a molecule called glutathione or GSH can cleave this disulfide bond in this polymer to release siRNA to function. So this is the overall rationale of the UMP design for siRNA delivery. And some of the principles, for example, the endosome escape, the active payload release mechanisms can be also applicable to designing the delivery system to for other biomarker molecules. And we found that this platform is very effective in inducing gene silencing. So we compared it with a commercially available liposomal-based transfection agent called the RNAi Max. So this has been used in many labs, and I think we found out that this the UMP platform actually lead to comparable gene silencing efficiency compared to the RNAi Max. and the RNMX is known to be cytotoxic to the cells. And because it's liposome-based agent, so it's also not stable. But the UMP system is more cell compatible and more stable, and can be potentially used for in vivo applications. So that's about this UMP nanosystems to deliver small drug molecules like the small drug and also the nucleic acids. So it can also be used to deliver the peptides. So for the time's sake, I won't elaborate this system. So you can see that it's a very stable, it's a very versatile platform. So, but then I sort of encountered a problem. So you may also notice as well that this platform generally is suitable for relatively small payloads. So for the large payloads, for example, the proteins or large nucleic acid like DNA, so the loading efficiency or capacity of using this UMP will be very low. So mainly because of this steric hindrance of these arms. So then I start to explore other potential platforms that are, while stable, of course, and can also deliver these big macromolecules like DNA or proteins. So here I'm going to introduce a system called the Nano Capsule and use this system to deliver CRISPR-Cas9 genome editor as an example. So a brief background of CRISPR-Cas9 technology, and I'm sure you heard about this very promising technology, and it's one of the most advanced technology in the field of genome editing, and it's also the most popular tool that being used currently in many labs, and it holds promise to treat otherwise incurable genetic disease. And once it's been demonstrated that gene editing capabilities in eukaryotic cells in 2013, within a short period of time, so this field has been explosively expanded with a huge market and research funding supports. And as well, and importantly, the Nobel Prize in Chemistry in 2020 was also awarded for the development of CRISPR-Cas9 as a genome editing tool. So two critical components in the CRISPR-Cas9 system. So one is Cas9 nuclease, so which can cut the double-strand DNA. And the other one is single-guide sgRNA, which can guide the Cas9 protein to a specific DNA sequence. So together they can form this complex called the ribonucleoprotein, or RNP for short. So although despite the great promise of this technology, so the biggest challenge of this this tool in the therapeutic applications is the lack of safe and effective delivery system. So this is mainly because the RNP, like the siRNA, is a negatively charged complex. So so it cannot effectively get into the cells by itself. So based on this negatively charged characteristic, so researchers have designed some positively charged polymer to deliver this RNP complex. So however, after a careful study on the surface charge distribution of this RNP, I found out that although the overall charge of RNP is negative, but it actually shows a very heterogeneous surface charge distribution. So the main negative charge domain is on this sgRNA chain, and the Cas9 protein, as you can see here, has both positive and the negative charge domain. So while in order to form a better delivery system for this RNP, so I'm thinking that using only positively charged polymer is not the best option. So I took advantage of this heterogeneous surface charge distribution and coat the RNP with a mixture of both positive charged monomer and negative charged monomer on this RMP surface. And here I also did include this imidazole monomer, so that it can help the RMP to escape from the endosomes to avoid the degradation of RMP, while the same principles as shown in the siRNA delivery, right? So, and in order to form a cross-link network to form a stable nanosystem, here I also incorporate this biodegradable cross-linker, and after simple in situ polymerization, so it can form this RMP cross-link nanocapsule around RMP and form this nanocapsule structure. And again, only covalent chemical bonds within these systems, which ensures its good in vivo stability. And to make it water soluble, so I also integrate a PEG monomer on the surface, and this PEG monomer can also allow us to integrate the target ligands to do the surface functionalization. So, like I said, so these nanocapsules can be taken up by the cells through endocytosis, and because of the imidazole group through a proton sponge effect, it can escape from the endosomes, and once in the cytosols, the polymeric shell can be cleaved and release RMP to do the gene editing in the nucleus. And since we have several monomers and also cross-linkers in this system, right? We carefully titrated and the monomer and screen the optimal nanocapsules formulations with the highest gene editing efficiency. So this is a busy figure, but importantly, I want to point out here is like we can see that it proves that the mixture of cationic and ionic monomer with the optimal ratio can induce much better gene editing efficiency compared to the use of pure positively charged monomer to deliver the RMP. And here is the TIM image of the optimized NC systems. We can see that it's quite uniform in size, and the average diameter is only about 16 nanometers in diameter. And we compare it with the commercially available lipovaccine system for CRISPR-Cas9 delivery that have been used in many research labs for in vitro lipobactamine is known to be cytotoxic to the cells, and NC shows a very good about cell biocompatibility. And more importantly, this nanocapsule system can be freeze-dried and maintain a robust gene editing efficiency after resuspension in the solution, which also indicate good stability, which cannot be, so this cannot be achieved by using the lipobactamine system. We also tested in some disease-causing gene loci. So here I just use the APP gene as example. So the mutations in APP are associated with Alzheimer's disease. So in the human embryonic stem cells, we can see that the nanocapsule system will lead to a clear gene edit in this APP locus. And so I mentioned about the surface functionalization, right? So here we want to see whether the gene editing degree can be further amplified by adding some target ligands or some, and here we use cell penetrating peptides, which can increase the cellular uptake of nanoparticles. So, and it can clearly increase the gene editing efficiency in this stem cells and in this APP gene locus. And we also tested this platform in vivo. So we test in a mouse model called AI14. So the uniqueness of this mouse model is that if you successfully carved the right gene locus, so those cells can express TD tomato red fluorescence protein, which can be easily detected through fluorescence microscope. So in this way, we can quickly identify whether our nano capsule system can work in vivo or not. So we test in several tissues. For example, in the eye, there's a retinal pigment epithelial layer. So the reason why we're interested in this cell layer is that many gene mutation happen in this cell layer and cause many eye diseases. And after sub-Q injection, so we examine the whole mouth. So we dissect the eyeball and look at the RPE layer carefully, and we can see that this nano capsule system, while clearly induce the gene edits as shown here, the appearance of red fluorescence signal, and by adding the ATRA, which is RPE. So RPE target ligands, so the gene editing efficiency was significantly enhanced as shown quantification here. We also tested in the brain tissue and found a pretty good, pretty robust gene edits in the primary neurons, which is very hard to do the gene edits actually, and we also show very high gene editing in the glial cells in vivo. So as a quick summary, so I designed this unique nano platform for intracellular delivery of RNP, and it's very stable and small in size, and it shows good biocompatibility and versatility in surface modification, and more importantly, it can induce robust gene editing efficiency in multiple cell lines in vitro and multiple tissues in vivo. So that's about the first part of my presentation about using nanoformulations for drug delivery. So now I'm going to switch the topic a bit to present you a microdevice-based drug delivery systems. And here the device is a microneedle array patch systems, and I leverage this system for enhanced cancer immunotherapy as an example. So a brief background of cancer immunotherapy, so which is to reboost our body's immune system to destroy cancer cells instead of directly killing the cancer cells like the chemotherapy or radiation therapy. And compared to the conventional treatments like chemotherapy or radiation therapy, so this emerging therapy has great potential to prevent metastases or recurrence. This is very important because it is estimated that 90% of cancer-related deaths are caused by the metastatic disease rather than the primary tumors, because many primary tumors can be surgically removed or using radiation combined with the radiation therapy or chemotherapy. So immune checkpoint blockade therapy is one of the most promising of cancer immunotherapy. And here I will just use the PD-1 and PD-L1 pathway as a example to introduce this ICB therapy. So cancer here, like cancer cells express their unique antigen on their surface, which can be recognized by the T cells, which is type of immune cells specific T cell receptor to recognize this tumor antigen. However, tumor cells will also express a protein called PD-L1. So this PD-L1 can bind to the PD-1 receptor on the T cells and send a don't eat me signal, actually a negative immune regulator. So it's like the tumor cells want to hold T cells' hands and saying, oh, we're family, we're friends, so don't kill me. So this is actually one mechanism that the tumor cells escape from our anti-tumor immune system. So the immune checkpoint inhibitor, for example, the anti-PD-L1 inhibitor or anti-PD-1 inhibitor, works by blocking this don't eat me signal sending by the tumor cells and reactivate the T cells to destroy the cancer cells more effectively. So of course, there PD-1 and PD-L1 pathway is just an example. There are some other negative immune regulators in this anti-tumor immune system that I won't introduce them for the time's sake. And currently, there are several approved ICB therapeutics for treating different types of cancers like melanoma or lung cancer, etc. Although the ICB shows quite promise in certain type of cancers like lung cancer or advanced melanoma, so the overall objective response rate of ICB therapy in patients is generally lower than like 40%. And for the most cases, the response rate is even below 20%. And also, ICB cause severe side effects because it allows the immune system to attack some healthy organs as well. So two possible solutions to overcome these two limitations is, for one, we can use local or target delivery system to deliver these immune checkpoint inhibitors to minimize this systemic side effects. And we can also combine the ICB with some other treatment method to synergize the ICB therapy. So here I engineered a device system that can locally deliver the immune checkpoint inhibitor. And this device can also allow us to combine the ICB with a treatment method called plasma therapy or plasma medicine. So while plasma therapy is here, the plasma here is meaning the ionized gas. So it's not the component in our blood. Okay. So it's a gas that composed of charged species like or radicals and photons. So it's actually the fourth state of matter which comprises over 99% of our visible universe, like the lightning or the fire we can see in our daily life. And this emerging technology have many potential applications in biomedical field, such as wound healing, decontamination, and more recently for cancer treatments. So what really makes plasma therapy workable in our human body is the generation of cold plasma, called atmospheric plasma, or CAP, which though the generate CAP is shown the purple here. So it's at room temperature and it's safe to apply directly on our body. So the mechanism of CAP to destroy the cancer cells is due to the presence of this reactive oxygen species and the reactive nitrogen species. So these two species can induce cancer immunogenic cell death in the cancer cells. And this cancer immunogenic cell death can cause the tumor-specific immune responses, which can potentially synergize with the ICB therapy. And the advantage of CAP therapy is that it only uses ionized gas without the administration of additional substances like the chemotherapy. We need chemo drugs, right? So that means that it could be a safer option to combine with ICB to enhance ICB. So in this project, I intended to combine the co-plasma with ICB, and I talk about the advantage of CAP, right? Certainly, there's some problem with this therapy is that because this species, these two species have limited tissue penetration. So we need a delivery tool for CAP to the tumor tissue as well, same as immune checkpoint inhibitors, right? So here now the clear idea is that we need a system that can locally deliver both immune checkpoint inhibitors and the CAP to the tumor sites. And here comes my design. So I engineer a unique holostructure. So this picture may give you a better understanding. So holostructure microneedle patch. So the idea is that I first apply this hollow microneedle patch on the skin. And the hollow microneedle patch can serve as a microchannel to guide the co-plasma through the skin to the skin tumor. Here we are targeting the melanoma. So the cap in the tumor tissue can cause the cancer immunogenic cell death, as I mentioned earlier. So this cell death can trigger the release of tumor antigen, which can trigger the following immune responses. And ultimately, it can induce the tumor-specific T cells, which can infiltrate back to the tumor tissue to recognize and kill the tumor cells. And the inhibitors loaded in this microneedle can be released in the tumor microenvironment. And for example, the anti-PD-L1 used here, it can block the don't emit signal and further potentiate the tumor killing effect of these T cells in the tumor sites. So, so to simplify it, so this local device can deliver both CAP and anti-PD-L1 immune checkpoint inhibitor. And the CAP can trigger the generation of tumor-specific T cells, and the immune checkpoint inhibitor can further enhance T cell killing efficacy. So this is set up to generate the CAP. So for the time's sake, I won't elaborate. Elaborate here. I will skip this. And here's the actual image of a CAP through the jet. So here we can see the CAP, and through the thermal imaging, we can see that it's really at the room temperature is around 22 degrees Celsius. And here is the proof of presence of the ROS and RS in the CAP. So as I mentioned earlier, CAP can cause immunogenic cell death. And to confirm this, we studied the generation of immune checkpoint, the immunogenic cell death marker in the melanoma cells. We can see that after CAP treatment, there is an increased level of this CRT, which is a ICD marker on the tumor cells. And this immunogenic cell death can further trigger the dendritic cell maturation, meaning that dendritic cells start to present the tumor antigen to the T cells. And here is the fabrication process of the Holostructure microneedle patch. So I used the process is actually very simple. So I used a silicone mold with multiple cavities and deposit the polymer solution on these cavities, and then just simply let the water evaporate. This microneedle patch can be detached from the mold, and this is the actual image of microneedle mold, microneedle patch. And this is the ICEM image. It shows that from the needle side view, we can see the formation of this microneedle needles. And from the base side view, we can see the clear observe this whole structure within each microneedle. And this is a 3D confocal image showing the holostructure of a single microneedle. And the structure is actually like the traffic horns, right? But it's much smaller in the micron scale. So then we want to ask whether this holostructure microneedle patch can really deliver the CAP, facilitate the deliver CAP through the skin, right? So we set up this experiment. So we have the CAP on here is the microneedle patch. We apply the CAP on top of the microneedle patch. And we clear and we monitor the species above the patch and under the patch. We can clearly see that there's a substantial amount of the RS and RNS after penetrating this holostructure microneedle patch. And compared with the solid microneedle, that means there's no holostructure in this microneedle patch. And also we test in the mouse skin, we can see that there's a minimum RS or CAP species penetrate through this solid microneedle patch or the mouse skin, while proves that the CAP have limited penetrating limited tumor tissue penetration. And once we apply the hollow microneedle patch on the mouse skin, we can see that the CAP penetrating efficiency has been significantly increased. And then we perform this in vivo test to see whether our hypothesis of combination combining CAP and ICB therapy works. So in this melanoma mouse model, we treat the mouse with different formulations, and I won't go over, but importantly, with this treatment, the hollow microneedle patch with both immune checkpoint inhibitors and CAP, we can see that it induced the best control of tumor growth and with a prolonged survival in this mouse model. And more importantly, so because our approach is local treatment strategy, right? We want to ask whether this local strategy can activate the systemic immune response to eliminate or to inhibit the growth of distant tumors. And here we treat the mouse. We have mouse with two tumors separate tumors, and we treat the tumor on the right side with this combination therapy, and we monitor the growth of both tumors. We can see that this treatment can not only inhibit the growth of primary tumors, it can also inhibit the growth of distant tumor, which has very high clinical impact because many distant tumor or metastatic tumor are hard to find or to detect or to be accessible. And as a conclusion, I use this local device to deliver both immune checkpoint inhibitor and for the first time, combine it with CAP therapy to achieve an enhanced ICB therapy. And I think I'm over time, but another short study is that we also apply this CAP for the post-surgical cancer treatment to inhibit the post-surgical tumor recurrence. So because the tumor recurrence after surgery is often seen in many cancer patients, causing the treatment failure, we can see that using the CAP therapy after at the surgical cavity to reactivate the immune system, it can inhibit the growth of post-surgical tumor growth. And importantly here, we design a portable CAP device which can provide a convenient strategy for complete clinical application of this CAP therapy. And here, essentially, we show that after tumor section, we apply the portable CAP on the resection cavity, and the tumor growth was significantly inhibited. So finally, I would like to summarize the topic that I've covered during the last around 15 minutes. So I've introduced two systems from the nanoscale to microscale as a drug delivery tools. And for the nano system, I focused on two stable nano platforms, Unimolecular Nanoparticle as a Nano Capsules, which can deliver various types of payloads for many applications. And I also explore other treatment methods like transdermal delivery approach using micro devices, and combined with some emerging technology like cancer immunotherapy or CAP therapy. And lastly, I'd like to thank my lab member, and also collaborator and funding sources. And the last slide, I promise. So my lab also have vacancies for 2 graduate students for the coming year. So interested candidates can send me your CV, or please spread the word if you know someone who may be interested. Yeah, with that, I would like to take any questions you may have. Thank you very much for your time.
Perfect. So thank you very much, Dr. Chen, for this very impactful talk. I think it delighted everyone in the audience. So the questions. So the first question is about the responsiveness, so that the release of the drug. As far as I understand, it basically is delivered in a stable form. But then, how does this drug get out from the carrier?
Oh, that's a very good question. So as I mentioned, so the drug carrier itself is stable, but the loading mechanisms, for example, if we use this unimolecular nanoparticles, so the drug loaded through the hydrophobic interactions or electrostatic interactions.
Mm-hmm.
So those drugs can be passively released from this payload because there's no covalent chemical bonds between the delivery vehicles and the drug payloads. And for this platform, so we have this polymeric coating, right? So it's biodegradable. So once inside cells, as I mentioned in the scheme, so it can be cleaved by the GSH molecule in the cells to release the payloads. So it won't affect the the payload release profile.
So more active strategies for the release, right? So yeah. the questions again. So I mean, to follow up from my side. So the pathology side could be very dynamic. And the conditions in health and disease are likely to change in a dynamic way. So does this design strategy also include the flexibility to adjust the carrier such that, I mean, those conditions, the likely changes are taken into consideration? So what's your attitude there?
Well, that's a very good question. I think that's also the future direction of this field, because while disease are changing all the time, but here essentially we have the way to engineering certain intelligent domain in this drug delivery system to respond to those changes. But I think still many there still needs some efforts to lead this field this direction.
So supermolecules are, I guess, promising, right? So they give some very great work. So the second question is about the HMN patch. So the question is, does its shape and aspect ratio matter?
Matter about the delivery efficiency? I think it's mostly the question about the shape and morphology of drug nanocarriers. And I do believe that the shape will change the delivery efficiency, but there's many research have been many research have been done looking at different aspect ratio, like a nanorods. So they're saying with the different aspect ratio, it may change the delivery efficiency, but my personal perspective that it's not the conclusion is not, the result or is not conclusive yet. So particularly still for those studies, they are still mostly in preclinical mouse model. So there lack of evidence in human clinical trials. So so I don't have an answer for that yet.
Thank you. So the next question is about the safety of CAP therapy. So what is your?
Yeah, so that's also a very good question. So the CAP, as I mentioned, so it's a room temperature. So and also it's a localized treatment strategy. So another big advantage of using CAP is that CAP can induce selective cancer cell death, which is the hypothesis is that cancer cells originally it has higher ROS level compared to the normal cells. So the lethal threshold of CAP by inducing the oxidative stress is easier to reach in the cancer cells, which lead to this selective cancer cell death. And we test the CAP on the dendritic cells as a normal cells. We found that at the same test condition, so the CAP treatment induced significantly higher cell death in cancer cells, but show minimum toxicity towards the dendritic cells, at least from our study.
Perfect. Thank you very much. So another question comes, from me. If there is any other question in the meantime, the audience can type. So it's about the interaction of these carrier systems with the immune cells and the complement factors present in the bloodstream. Have you looked into the potential interactions in the short and long time scales? Is there any contribution from the to the instability of these carriers from the complement factors, so the immune system gets alerted, or any sort of observation in that part?
That's a very excellent question. So I didn't do much research on this drug nanocarriers, their interaction with the immune system, but here we mostly use very biocompatible polymers, and also some FDA-approved polymers, which shows the good biocompatibility. But I agree that these polymers still these foreign materials, right? It can trigger some immune response. For example, there's many evidence showing that the PEG polymer, which has been used very widely in the field of drug delivery, it can still trigger some immune response and generating some antibody towards these polymers. But still, so that's add another limitation to the field to use the drug nanocarriers as a drug delivery, right? So that's why I'm thinking that the stable is very important, because while the interaction with those cells and also the serum proteins, antibodies, can disrupt the structure of nanoparticles. That's why the stability is very important, even in presence of immune system or immune cells.
Yes. So, are there any questions? So, if not,
I have a question, but in case any audience also have questions, you can raise your hand if you want to be promoted to panelists and ask it live, or type it in the box if you want us to read out. So my question is on what is the similarity, difference, or potential advantage of your system over viral vectors, which is kind of a growing field in industry right now?
I think that's an excellent question, and I think I didn't emphasize too much about this. For example, the CRISPR-Cas9 delivery system, right? Here we use non-viral delivery systems. So the biggest advantage of using non-viral system is to eliminate some immunogenicity or insertional mutagenesis associated with the viral vectors, right? That's why, for example, the COVID vaccine, so like Pfizer or Moderna, they're using the lipid nanoparticles to deliver mRNA, although the some other vaccine also use viral vectors, which is considered as a AAV viral vectors, which is considered with a good safety profile in humans. But there's still a problem with the viral vectors. For example, the viral vectors cannot deliver large payloads. For example, the CRISPR-Cas9, which is a limitation. So you have to you cannot use AAV, which is a safe viral vectors. You have to use lentivirus or some other viral vectors, which is not being demonstrated or still have some safety concerns, right? That's the advantage of using non-viral vectors. The safety for the safety aspect. And but while the limitation of non-viral vector is the efficiency. So compared to viral vectors, so the efficiency is very low, or is low. So that's why we are engineering some intelligent delivery approach, targeted delivery approach to increase the delivery efficiency or transfection efficiency, but with minimized safety concerns.
Perfect.
Cool. Amazing. We didn't get any more questions. No? Okay.
Yeah,
I have another one. Maybe could be the final one. On the ligands, so are the ligands now designed to be specific to the tissue type or tumor type? Is there potential for it to be more personalized, i.e., the patient's specific biomarkers in the disease tissue?
That's a very good question. So I think the target ligands is also a key factor that influences the drug delivery efficiency if we're using nano platform, right? So I think it depends on different disease, right? For the cancers, different cancer overexpress certain receptors, and target ligands can. Well, my personal perspective is that target ligand can only increase. It cannot, showing the 100% delivery efficiency to the tumor tissue. To me, I think that's impossible at this stage. So the target ligand can only increase the delivery efficiency. And I think compared to the free drug, that's usually like less than 0.1 or 0.01% to the cancer type, using our nano platform can obviously increase the delivery efficiency, although it's not optimal at this stage, but I think generally it can, the delivery efficiency is much higher compared to the free drug formulation. And in terms of personalized medicine, I think while cancer immunotherapy is a great way, because while it uses the tumor antigen, right? It's generated specific tumor antigen. And the advantage of using the approach that I designed, for example, the CAP, to generate tumor-specific antigen in situ, so without considering the heterogeneity in the tumor type, because different stage. So in this way, we can include many type of tumor antigen in using this approach, instead of using, for example, some other strategy using the in vitro engineered immune cells, which can have some limitations in terms of the tumor antigen or neoantigen you can engineering or design. But if you can in situ, like generate the tumor antigen, that could be a better way for implementing the cancer immunotherapy.
Cool.
Okay, I hope I answer your question.
Yeah, thanks a lot.
So yeah, if there is no other question, then we can close the session this week. And yeah, thank you very much for everyone who joined today for Dr. Chen's seminar. And Dr. Chen, thank you very much again for taking your time to share your amazing work with us, and hope to interact with you more again in the future.
Thank you.
Thank you.
Bye-bye.